for this conference. So Dr. Jean-Marc Schwartz is a senior lecturer in the division of evolution in function and genomics at the University of Manchester. He leads research in computational system biology, specializing in metabolic network modeling, high-throughput data integration, and data modeling of cellular pathways. He earned his PhD in computer engineering from Laval University in Canada. So now it's to you, Jean-Marc.
Yeah, thank you, Sabine, for the introduction and for the kind invitation. So now I will share again. Okay, I suppose you can all see it now. Yeah. Okay, so the work I wanted to present today is relatively recent and still in progress, but I hope it will be of interest to so many people here. So it's a project that was mainly conducted by Kate Misson, who's probably also listening at the moment, so she can perhaps help answer questions at the end if I can't. And it's also, yeah, and this work has not been published yet, but has been presented at the Metabolic Pathway Analysis Conference this summer. So if anybody attended the conference, this will sound familiar to them, but hopefully not everyone attended the conference already. So okay. So before I start presenting, first a little bit of background. I prepared this slide on flux balance analysis, so I suppose many of you are familiar with what FBA is, but just in case some people are not familiar. So flux balance analysis is a technique that is widely used in order to calculate metabolic fluxes in metabolic networks. And it's mostly used in genome-scale metabolic networks. So these are large models which contain all the known metabolic reactions for organism or cell type. So the way FBA works is that we represent metabolic reactions in the shape of a stoichiometric matrix, which you can see an example here in B. So stoichiometric matrix is a big table where we put all the reactions in columns and all the metabolites in rows. And then we enter the stoichiometric coefficients in this matrix. Then we apply mass balance equations. So we assume that we have a quasi-steady state, which means that all the incoming fluxes have to be equal to all the outgoing fluxes. Or another way to see it is that there is no accumulation of internal metabolites. So the sum of all fluxes that produce a certain metabolite, internal metabolite, has to be equal to the sum of fluxes that consume this internal metabolite. And then there is one more crucial step, which is to define an objective function. And the objective function represents the combination of reaction fluxes to be maximized. And it is then maximized using a linear programming algorithm. So essentially, this little panel E here summarizes how it works. So we have the previous constraints, so all the mass balance constraints and some, for example, maximum capacities and so on, that determine a certain solution space shown in blue. And then once we defined an objective function, we are looking for the optimal of this objective function, and that's the result then of FBA. So the question is, what exactly does the objective function represent, and what type of objective function is suitable for a given problem? And this is actually a relatively complex question, which depends a lot on the context. So in many, let's say in many biological studies, people have a tendency to use biomass production or growth as the objective function. So this, I would say, we could say describes a kind of natural environment case where we assume that species have evolved in the environment in order to optimize their growth, and then they behave in that way. They continue to behave in that way. But that's not a universal solution. So it could depend on other factors. For example, if a species, for example, is suddenly put in an environment where resources are more limited, and it cannot it cannot use essentially all the resource, it cannot find all the resources or all the compounds it would need, then maybe it could switch to another mode, which would be to preserve its metabolism rather than optimizing growth. Or, for example, if we have more complex organisms, it is very likely that also the objective depends on the cell type and maybe also on the cell cycle and different other factors that can affect what objective is actually meaningful at a given time. If we look from the modeler's point of view, well, an objective function is a set of reactions to be maximized, which enables FBA to find a single or at least a more limited set of optimal flux distributions. I just found it worth noting that recently this year there was an article published that tried to characterize different types of objectives, and they defined type 1 objectives, for example, would be now something like engineering goals. So that's another context where objective functions can be used. Because, for example, if we do biotechnology, then we don't necessarily want to use a natural objective, but we would, for example, look for ways to manipulate or transform an organism in order to optimize the production of certain compounds that have commercial or biotechnological value. So in this case, the objective is actually tweaked towards what we want to see the cell doing. So that would be type 1 objective according to this new definition, and type 2 objectives would be more things like hypothesis-driven objective. For example, we assume that the cell maximizes its growth or maximizes its ATP production or other objectives like this. So yeah, just to locate the context of this project. So this work, we actually developed it as part of a larger project, which was financed in part by Fujifilm, and which deals with Chinese hamster ovary cells. So essentially, they are using CHO cells in an industrial context in order to produce monoclonal antibodies. And essentially, they are, of course, interested in maximizing the production and the quality of the antibodies that are produced. So essentially, the idea is, can we use metabolic modeling and genome-scale metabolic models in order to find ways to optimize the culture process or propose some modifications that can help improve the production of monoclonal antibodies? And in this case, of course, we are dealing with mammalian cells, so things that are something that is much more complex than a unicellular organism, and therefore the objective function is not that obvious. At least it's not clear that it should necessarily be growth that these cells are trying to optimize. Another piece of background is that this question is not new. This has already been approached by some other people before. Here are just a few examples. So there was actually in 2016 this paper you can see on the top left, which was introducing something called inverse FBA or INV-FBA. So what they did was essentially to take the FBA problem to inverse, well, essentially the FBA problem. So rather than defining an objective and then calculating all the fluxes that result from it, they started from a set of fluxes and then calculated the set of objectives that are compatible with this output. So essentially they reversed the linear optimization problem. The problem with this approach is that it gives us very complex. So first, it gives a very large set of objective functions and also a very complex set of objective functions. So they are not necessarily all meaningful in a biological sense, and also they're not easy to use then in order to do future applications. There was this more recent article which introduced something called SCUTI. So in this case, they need omics data. So they took an approach using omics data and machine learning in order to try and infer metabolic objectives in single cells. Here again, it's a very nice work, but it's based on omics data, so we don't necessarily always have omics data for the problem we want to resolve. And there's also this paper by Nathan Lewis at the bottom. So in this case, they took a more systematic approach. So they essentially tested lots of different objective functions, so mostly in the context of CHO models, which is why we cited it as an example here. So they kind of more systematically tested all the meaningful objectives and tried to find which one matches best the measurements or the data they had. And essentially one of the important outputs was that indeed growth maximization is not really the best objective. So to find something meaningful, we need something a bit more complex than just using growth. So our aim was to try and make this process a bit simpler by developing a Python tool that is aimed at predicting the best suited objective function for a given simulation. So again, yeah, the idea is that if we don't use the right objective function, then essentially our predictions made using flux balance analysis could be very wrong or essentially useless. And also, yeah, why do you want to do this? Yeah, that's what I was saying. And also the other problem is that if we look across the literature, so this is just a review done by Kate, again, across many different articles that presented that used FBA for different types of organisms and to see all the range of different objective functions that were used or that can be used. So you can see that biomass production is usually the most widely used, but there are lots of other possibilities like ATP production, maintenance, minimizing enzyme costs, minimizing the sum of intracellular fluxes. This is the technique called PFBA and lots of others. So essentially, so it doesn't mean that necessarily some of these are wrong and others are right. It means that it all just depends on the context and that we cannot actually probably not have, there is no such thing as a universal objective function that could be used across all situations. We really need to find the best function for a given context and problem. So in the case of CHO cell cultures, this is particularly important because if we look at how the CHO cells how they grow and how they produce antibodies in culture time, we can distinguish three main phases. So it usually starts with something what we call the lag phase, where initially the growth is quite low, so the cells are still adapting to the culture conditions, and the production of antibodies is also low. And then in the middle, there's the exponential phase where then the growth becomes very high, but e.g., so antibody production is still quite small, but increasing. And then finally, there is the stationary phase where cells start to die, and so the growth rate drops, but IgG production is very high. And essentially, so again, in a biotechnology context, what we would like is actually make this final phase as long as possible because we want to produce as much antibody as possible. And of course, other types of cells can have similar or different phases where the objective or actually the characteristics of growth and production change over time. So this result shows why it's absolutely crucial to have the right objective function. So here are just three examples. So if we use, so on the left, you can see if we use growth maximization, we predict by FBA a certain amount of growth, which is about 0.002 here, and a certain amount of IgG production. But then if we switch the objective to IgG production, then there is a bit more of IgG production, but there is no growth at all. So this is not very realistic either. And if we use 50-50 split objective, so which means combination of growth and IgG production as the objective, then we have something that looks a bit like growth maximization. So we can see that the choice of objective function absolutely determines the result of the FBA simulation. Another way to look at it is to look how, for example, the objective function controls gene essentiality, because this is another common problem. So if we want to, for example, look at transformations, cell transformations, in order to optimize the cells to a certain objective, we often try to do gene deletions or sometimes insertions, but mostly deletions. And here again, the results of gene deletions and gene essentiality very strongly depend on the choice of the objective. So on the left, you can see, for example, that if we use growth maximization as the objective function, then most of the genes are orange, which corresponds to reducing IgG formation only. So it means that there are lots of targets that reduce IgG formation, which is not what we want, essentially. And there are just a few that reduce both growth and IgG. These are the green ones. If you look on the right side, then most of them are red, which means no effect. So the gene deletion doesn't affect neither growth or IgG formation. And only in the middle, you can see that there is a little blue part in the pie chart, which corresponds to genes that reduce growth only. So these are essentially the ones that we would like to have. So genes that, when they are deleted, reduce growth only, but do not reduce IgG formation. But again, you can see that only if we use here a 50-50 split objective, we can find such genes. And if we use growth or IgG at 100%, we cannot find these genes. So again, the choice of objective function completely determines the output of such a study. So the question, of course, is what is the right balance? How can we find something that's actually most realistic? So here we are then coming to the solution that we proposed. So the idea was to develop a function that acts as an objective function finder. And we started by developing it against well-characterized single-cell organisms like yeast and bacteria, and testing it across a variety of conditions. And then in the end, we will apply it to mammalian cells and as a validation, check whether we can better predict gene essentiality using this fitted, so this predicted objective function rather than using an arbitrary objective function. So the algorithm itself is based on an algorithm called simulating annealing, which so again, not sure if everyone is familiar with it, but basically it's a sort of analogy based on the physical process of cooling a big piece of metal in metallurgy. So the idea is that, well, I think the next slide explains it a bit more. Yeah. So the idea is that we want to find an optimum in a complicated space. So we start with a sort of arbitrary solution, and then the algorithm will randomly mutate or randomly change the initial solution by changing some of the values randomly, and then check if the new solution is better than the initial one, then it is accepted for the next step. But also, even if the new solution is not necessarily better, it can be accepted with a certain probability. And the advantage of this is that this algorithm is quite good at escaping local optima. So, for example, if the algorithm was actually falling into this kind of local optimum, which is actually not the best optimum, it still has a chance of leaving this valley basically and going back into finding the real optimum. And so the probability that we can actually go up again decreases over time. So this is the analogy of the temperature in this annealing process. So essentially the temperature is very high at the beginning, which means at the beginning we can explore a very large range, a very large space. And then as time progresses, the temperature is gradually decreased so that towards the end, the exploration will be much more local. And eventually we hope that we find the best solution. So this algorithm particularly was chosen because it's quite good at finding, at escaping local optima. So it's quite good in the case where there are kind of multiple optima and we have a quite complicated search space. So the way our algorithm then works is that the user will need a COBRA model or a genome-scale metabolic model, which can have some enzyme or media constraints. And then importantly, we chose to base it on qualitative criteria. Because again, the idea is to make it practical and easy to use. And in many cases, when we want to do FBA simulations, we don't have very detailed set of fluxes for many reactions, but we know what happens qualitatively. So we know that some reactions, some nutrients are taken in, some things are produced on, and some reactions and some inputs are, for example, blocked. So rather than using quantitative numbers, we chose to have qualitative numbers for the kind of input constraints, which will be -1, 0, or 1. And then we need also to define a set of, let's say, candidate objective functions. So rather than using every possible reaction, which is not something that's practical in a genome scale model, we define a limited set of candidate objective functions, so the key reactions that we usually know are meaningful. And then the output of the algorithm will be then a suggested suitable function that combines, that can be, for example, a single objective or a combination of several objectives. And it will also show us what is the maximal accuracy achieved. So do we match 100% of the criteria that we set in the inputs? And also we can check intermediate searches and see how the algorithm found that solution. So here are some of the results. So as I said, we wanted to develop it first using unicellular organisms. So that's then most of the development and validation was done initially. So here we initially applied it to yeast, and so this was done actually as part of a collaboration with Bas Teusing's group in Amsterdam. So they helped us to define two main phases or two main types of growing yeast cells. So slow-growing yeast, which we call here slow-growing yeast, and fast-growing yeast. So it essentially depends on the amount of glucose they have. So in the slow-growing yeast, we have, when in both cases, we have yeast that is actively growing, which is why biomass production is set to 1. They both take glucose in, so glucose uptake is set to -1. But the difference is mainly in the ethanol and glycerol exchanges, which are 0 in the slow-growing yeast and 1 in the fast-growing yeast. So they start secreting ethanol and glycerol in the fast-growing phase, but not at the beginning when they have minimal ethanol and glycerol exchange. And also there is ammonium uptake, so this is then set to -1. So these are then all the criteria that we want our simulations and our objective functions to meet. So first, applying our algorithm to the slow-growing yeast case, we can see that the solutions are relatively easy to find. So basically, there are 3 possible solutions, which are all good. You can see, so they are the ones shown in green on this slide. So we have growth maximization, we have glucose minimization, glucose uptake minimization, and we also have ammonium minimization. So either of these objective, so essentially what the green color here means is that the solution calculated by FBA matches the experimental qualitative criteria that we set as conditions. And you can see they are all green for these 3 objectives, which means each of these objectives would be suitable to represent this condition. And to complement this, we also tested what happens if we make combinations of these objectives. You can see that on the table at the bottom. So if we test an objective where we have 1/3 of growth, 1/3 of glucose, and 1/3 of ammonium minimization, again, it works. All the criteria are correctly met. So in a way, you could say in this case, there's no real need for our algorithm. You could just use a simple objective and you get good results. But things become much more complicated when we have the fast-growing yeast. So here we need a much more complex objective function, and we can see that actually none of the simple objectives work. So here, again, green means that the model, so the output of FBA matches the experimental condition, and red means that they are not matched. And you can see that for each of the 5 possible single objectives, there's never a completely green line, which means there's always at least 1 or actually even always 2 criteria which are not properly met if we choose this objective. And same thing if we use again a simple combination where we combine all the 5 potential objectives in a combined objective function with 0, so with 20% of each. Again, we have 3 green ones, but also 2 red ones, which means that again, in this case, growth is not maximized because it's predicted to be 0. And also glycerol is not maximized because it's also predicted to be 0 in this case. So no intuitive objective function can accurately predict these metabolic properties of higher growth yeast. And then that's why then in such a case, we tested our algorithm and see that in this case, we were actually able to find a solution. So this is again now our own algorithm applied to both problems. So on top, you have again the slow growing yeast. At the bottom, you have the fast growing yeast. And the colored cells essentially shows you how the algorithm actually looks for solution over time. So you can see that at the beginning, it needs some time to explore some solutions which are not good, but then eventually after a certain amount of time, it will find a solution that matches all criteria. So at the end, everything is green. So it's interesting to note that in the slow-growing yeast, the solution is found much faster because we've seen that it's an easier problem. So after about 50 iterations, we already have a solution. In the case of the fast-growing yeast, it takes more time. So here it took us 225 iterations, but still we find a suitable solution in the end. So then it's interesting also to look at what this solution or these solutions look like. And so here again, so it's worth noting that essentially like FBA, there is not necessarily a unique solution for the objective function. So we can actually find, so if we apply this algorithm several times, we will not necessarily always find the exact same solution. So what we're showing here is essentially the range of coefficient values that give a good objective function in these two cases. So you can see that on top, for the slow growing yeast, there is a kind of wider range of coefficients that can be suited. But in the case of the fast growing yeast at the bottom, actually the range of suitable objective coefficients is quite narrow. And you can see that it always ends up with about so something like, yeah, 90%, more or less 90% of growth and a small amount of ethanol exchange and glycerol exchange, so about 4-5% of each of the two others. And this is, so this is another way to look at these results here. So we also tested all the different combinations of these 5 objectives. So here, so this upset plot basically shows you, so at the bottom you can see all the objectives tested individually or in combinations. And you can see that actually only the one that combines ethanol, growth, and glycerol is the only one that achieves 100% accuracy. So all the other combinations of objectives were not able to meet all 5 criteria that we defined, maximum, so usually 3 or maximum 4. And one more way to look at these results is also to look at how the objective, so the coefficients of the different objectives in the solutions are correlated to each other, and we can see that there's actually a very strong correlation here between ethanol and glycerol exchange. So essentially they are kind of, they have to be at kind of similar level, but both are also very strongly anti-correlated to growth. So essentially if growth goes up, ethanol and glycerol secretion will decrease. And so yeah, so I think time is almost over and I've almost finished. So this is so therefore this is kind of the state of our work so far. So in the meantime, we've already continued applying this algorithm now to CHO cells, but this is not yet completed. So at this stage, we can say that we found that, so essentially what these examples that we've done so far show us is that in, let's say, relatively simple cases, simple intuitive objectives can be sufficient. But if we have several combinations of exchanges that have to be met, it's actually very difficult to find a single objective function that matches all the conditions. And we usually need to use a multi-objective function. And in this case, it's a good approach to use this sort of algorithm to find a suitable multi-objective function. Moreover, I would say that as we've seen in this case, the range of actually good solutions is very narrow. So we are quite unlikely to find it if we just kind of test randomly or arbitrarily different combinations of objectives. And yeah, so as I said, the next step will be to apply that to CHO, but that's still in process, so I'm not gonna insist on it today. And essentially, yeah, so some possible points for discussion of future work. Well, one idea would be, would it make sense, for example, to use a continuous instead of a discrete input criteria? So this has, well, this could have some advantages because if we have measurements, we may want to find a solution that's more actually narrowly, that more precisely matches the measurements we have. But on the other side, we don't always have precise measurements. So the qualitative criteria are actually more widely applicable. So there may be also other ways to do that. For example, using Markov chains algorithm, but I would say so far the tests, at least maybe Kate will correct me if I'm wrong, but I think so far the tests that have been made show that simulated annealing really seemed to work best for the examples that we tested. And of course, an alternative to this is also to use flux sampling, which enables you to check actually wider ranges of flux distributions without having to define an objective. But this is not always feasible for very large model and takes very a lot of time and resources. Yeah, okay, maybe I'll show that during the discussion. Just moving to the conclusion first. So thanks to well, Fujifilm, which partly funded this project, and we have collaborators in Edinburgh, Manchester, and York. And also particularly thanks to Kate Meeson and Rachel Gaffney, who did most of this development. And yeah, and then I just wanted to add that. So that's another thing that might be useful for some of you. So that's not related to this project, but we also have some material that Kate has developed that introduces constraint-based modeling and FBA for students and has several tutorials for yeast and CHO and human cells. So if anybody finds it useful, please scan the QR code. And that's something we found very useful because we have new students arriving all the time, and it's always difficult to restart explaining them, introducing people to FBA and this sort of techniques. So I think that can be a useful resource for some of us that work with students. And thanks for your attention.